I'm really excited to be here and not only show you some of the work from our lab, but also have a chance for discussions with people throughout the rest of the day. So I think it'll be a fun day. And thanks, Mishka, too, for organizing everything for the day. So as a lab, we like to think about how cells respond to their environment. And the reason for that is ultimately to be able to manipulate these systems towards some sort of therapeutic end. And the way we think about this is shared by many other groups, but we can sort of conceptually organize a lot of this into sort of 3 different layers of the things that we care about. So to sort of simplify and organize the parts of the system here, we can think about cues, and these would be things that cells are responding to or things that we're adding to the environment to somehow manipulate the response of cells, whether that's growth factors, drugs, or the extracellular matrix environment. There's these signals, which are the things that we can measure, but aren't necessarily directly influencing. And cells use a whole coordinated set of molecular factors to change their behavior. And then there's the responses. These are the things that we're generally going to care about. We can measure them. And what we spend a lot of our time thinking about is how do we build models that use the cues and the signals and relate those to the responses so that we can maximize whatever sort of goal that we have in manipulating these cell behaviors. Now, something that I think the best way to describe it is that we've struggled with this as a lab for quite a while is that there's just so much complexity here. And in fact, when we start thinking about multicellular systems, this is even more of a mess. So we can think about there being multiple cues. And then there's different sets of cells. These can be different cell types. These can be heterogeneous groups of cells. And each of these signal and therefore respond in slightly different ways or share common patterns of regulation. And then finally, sometimes we may care about multiple different responses. And so what I'm really going to try and show you today is we have, I think, some solutions in thinking about this. And I'll introduce some of the terminology in a bit, but really what this comes down to is thinking about these as separate modes. So thinking about these as separate dimensions for how cells respond. And so I'm going to kind of label this up front that we can think about cues as being mode 1, signals being mode 2, and responses being mode 3. And if we do that, we're going to have a great way of starting to organize this and make it easier for us to find patterns in these kind of responses. Now, why do we need to think about the fact that cells are really highly dimensional? It's because this creates a really huge space of different responses that we can see. So I'm sure that many of you could come up with lists of 10 growth factors you might want to treat your cells with and see how the responses are similar or different. You could try that in different extracellular matrices. You could try it with different dimensionalities, different drug treatments. And you quickly end up with millions of combinations that might be possible here. And that's just not experimentally tractable. And it's even sometimes not computationally tractable. And it's certainly not possible to explore something that we collect across all of these dimensions. And so we need ways of being able to find the patterns among these overwhelming numbers of combinations. And all of the methods that I'm going to talk about today are dimensionality reduction methods. And I like to talk about dimensionality reduction as being sort of a kind of magic. So it makes everything better. It helps us with visualizing data. It helps us with reducing data down, actually making it smaller so that methods that otherwise wouldn't be possible can now work. If we find the appropriate patterns in our data, so for example, there's this S here, and if we can identify that, we can actually we can remove things that we don't care about in our data, be it noise or batch effects. And if we're missing values, we can actually figure out what those missing values are by using dimensionality reduction methods. And some common methods that you may have used before would be something like principal components analysis or non-negative matrix factorization. And the way that these methods, or many of these methods, work is by finding what would be called a low rank approximation of your data. So you have some giant matrix with a bunch of different measurements in it, and we try and find what are the patterns across the features that explain the data, and what are the patterns across the samples. So how are the different treatments maybe related to each other? And then we can summarize that through the set of PCA, these would be principal components, the set of values that explain those feature to feature or sample to sample patterns. Now, to make this work better for these more complicated data sets, we're going to try and generalize this process to something that's a little bit less common, but it's going to have some really nice, useful properties. So to introduce some of the terminology here, if you have a single number, you have something that could be called a scalar, or another way of referring to this would be a zero-mode tensor. A vector of numbers, just a line, would be a one-mode tensor. A matrix is a two-mode tensor because there's a grid, two-dimensional grid of numbers. And then we start to get into some new things. So a cube of numbers would be a three-mode tensor. A hypercube is a four-mode tensor. So, we usually think about organizing our data in matrix form, but if we have measurements across different dimensions, we have something that naturally might fit into a 3D cube of numbers, or something that I have trouble visualizing. And then I'm going to use these terms. If you take your 3D cube of numbers and you maybe stuck like an apple core through it, so you pulled out a single line of numbers, that's going to be a chord or a fiber. And if we think about like slicing with a knife off the top of this cube, then we're going to end up with a slice. So if you pull out a vector or a matrix from some larger set of data, then you're going to get these two different terms. And so there are dimensionality reduction methods in tensor form. So this is one type of method called canonical polyadic decomposition. It's a scary name for what is essentially PCA in higher dimensions. And this, just like PCA, it's going to find patterns in the data, and it's going to give us a matrix that explains how the patterns are related to each of the dimensions. So how the patterns are related to the rows, the columns, and this new dimension that's depth. And one interesting aside, I'll show some examples so that you can see how we can easily interpret these patterns. But tensor decomposition is actually different from how we do matrix decompositions or a dimensionality reduction, because there are actually different ways that we can set up this problem. So this is one way, and this is a way that I think is really powerful, but there's additional flexibility we have when we're doing this in higher dimensions. So I'm going to give you the point up front and then show you a few examples of how we found these methods to be incredibly useful. And really up front, the unique benefits of doing dimensionality reduction in tensor form is we may actually be able to reduce the data down better than we would be able to do with PCA. So we're actually more effective in shrinking our data set and finding patterns that are simple to explain. One useful thing that's kind of difficult to recognize at first, but we'll see in some examples, is that even if these methods are just as good at reducing your data down, one nice property is that they assign patterns to a specific dimension or mode. So we're able to say, if we have, say, a 3D grid, data grid, where we have treatments, time, and cell types, we can say exactly what is the effect of treatments, accounting for all of the different cell time points. So it makes it easier for us to say, what's this effect of this particular thing that I'm manipulating? And then finally, because effects can be explained on a specific mode, these really naturally work for data integration. So if I have, say, measurements of different types in the same people, I can figure out what are the patterns across both of those data sets. Now, these aren't going to solve everything. They're mostly going to work when there's mode-specific effects. So what I mean by that is if a treatment has a similar effect across different cell types or across different time points, then organizing your data in tensor mode may be useful. So I want to show kind of 3 vignettes of how we've used these methods and work on showing it on different scales each time. So first, I'm going to show how we've used these to figure out what are the patterns of responses in immunocytokines. We'll work a little bit higher level and think about if we have not just one drug that we're trying to optimize, but we have a population of molecules that are all slightly different, how we can actually explore patterns in that data. And then finally, I'll show you how we can make this really complicated and look at multi-omic signatures in patient data and figure out what are the determinants of a disease outcome. So just thinking about one molecule, we've been working for a few years now and thinking about how we can build models of the IL-2 pathway and cytokine pathways, because this is a really nice system to be able to look at how responses are similar or different between cell types. So IL-2 is a cytokine that leads to modulation of the abundance and the activity of different immune cells. And its most prominent effects are in expanding or modulating the activity of T cells and some other effector populations. And that's led to a lot of interest in designing either IL-2 itself or variants of IL-2 that have effects that can be therapeutically useful. IL-2 has an alpha receptor, which is very high affinity for IL-2, but doesn't have an ability to signal. And then it has a lower affinity receptor, the beta gamma receptor complex, that it has the signaling capacity. And through just this seemingly fairly simple receptor family, this leads to a lot of complex effects across the immune system and seemingly contradictory effects. So just to kind of overly simplify how IL-2 works and why this has been therapeutically useful, IL-2 is actually an approved therapy in melanoma. And the reason for this is because it can selectively expand a lot of effector populations that are pro-inflammatory. So this would be CD8 positive T cells, NK cells, helper T cells. And this can be really beneficial for an anti-cancer response and anti-cancer responses in combination with other treatments like anti-tumor antibodies. And so there's been work more recently now in trying to actually make this signaling more biased towards the beta gamma receptor by eliminating the alpha receptor binding, and so that we can selectively expand these cell populations. Now, why do you want to get rid of the alpha receptor binding? Well, the reason is that if you use IL-2 at low concentrations, or you use variants of IL-2 that have more alpha receptor binding, you get selectivity towards Tregs, and you get a dampening of this anti-cancer or otherwise inflammatory response. So this shows kind of the back and forth of how IL-2 works, that in one extreme, you have this pro-inflammatory response, but with the same molecule at just a different concentration or a different binding affinity, you get seemingly the opposite response. And so this has inspired a lot of new ways to try and make these molecules more selective so that we can maximize these effects. And IL-2 in this regulatory T cell specific form is being investigated in a lot of different diseases, but examples would be in lupus or in transplant rejection. So this has, again, inspired a huge variety of different approaches in how to design these molecules. There's bispecific IL-2 fusions, where these IL-2s may have the wild type affinity of IL-2, or they might be muteins that bind to the receptors in a different kinetics. There's monovalent fusions. There's immunocytokines, where these IL-2s are bound by the antibody Fab region itself. And then there's this interesting company that's actually pegylating IL-2 to block or inhibit receptor binding and bias its signaling again. But again, all of these methods, ultimately their goal is to modulate these affinities. So have a more inflammatory effect through beta gamma receptor binding, or an inhibitory effect through alpha receptor binding. I'll show you the in vivo data up front to kind of motivate this. So this is great work in collaboration with Jamie Spangler and Derek Van Dyke, actually out at Hopkins. And they did this really detailed characterization of IL-2 immunocytokines, where they've modulated the affinity in a huge variety of setups so that we can actually see the trade-off that we have here and the limitations. So if you have an IL-2 that binds, say, to only the alpha receptor, you're going to get the maximal specificity for regulatory T cells, but you don't actually have a drug that does anything. Because again, the alpha receptor is completely signaling incompetent. Now, if you start to add some signaling towards the beta receptor, you start to get Treg specific signaling. And this is showing expansion of different cell populations when we treat with these immunocytokines, and then we've helped to visualize what these patterns are. But you finally get to this trade-off where you stop having this principal component 1 that explains this expansion of Tregs, and eventually you switch to only increasing along this other dimension. So you switch to expanding effector populations, but not Tregs anymore. And what's interesting is that they found that the optimal molecule was this one shown in purple. That was what they started with. And so this shows that there's a limitation fundamentally with affinity modulation, in that if you have something really specific, it doesn't signal at all. If you have something that signals a lot, it's not going to be specific. So we were wondering how we could do better. And so in collaboration with a biotechnology company, Vistera, we made a panel of 13 different IL-2 variants. And we tried to see if there's other things that we could vary to get more specificity. So these variants have IL-2 fusion in different orientations, either at the bottom or the top of the fusion. We have monovalent or bivalent IL-2 forms. And then we have muteins that have varying affinity. And if you do this profiling across 12 concentrations, 5 time points, 8 cell types, you end up with something that looks a little bit like this. And this isn't to show that there's anything that you can really extract from this. But I want to show that one of the limitations or the difficulties that we had is that even if you throw this into something like PCA, it's really not possible to figure out what the patterns are here. So every row here, and this would end up being your scores plot, is a combination of a concentration and a mutein. And every column is a measurement that was made at a specific time point for a specific cell population. So even if you reduce this down, you end up with these effects kind of combined together. You don't really know what the effect of the molecule versus the concentration is. And we'd really like to be able to separate those out. So instead, what we did is we organized the data in this 4-dimensional structure where we have cell types, ligands, time, and concentration. And each position in that 4-dimensional structure is a measurement that we made. And you can use CP decomposition then to find that there's roughly 3 patterns in the data. And 3 patterns explains about 90% of the variance. And we're able to go from about 4,400 measurements down to 100 values. So we're greatly reducing this data down. And then what's really important about this is that we can very clearly figure out what the patterns are here. So this is showing what those 3 different patterns are, and I'll kind of walk you through how we can interpret this. So I'm really going to mostly talk about component 2, which is the second pattern in our data. And what we noticed about this is that it's maximized at intermediate concentrations. It doesn't really have dynamics that are any different from the other patterns in the data, but it's extremely selective towards regulatory T cells. And so we thought, well, okay, this component 2 is really important to see which molecules are maximized in their component 2 signaling while minimizing the components 1 and 3, because what we're desiring to do here is to maximize the signaling in Tregs. Now, what was really surprising to us is when we looked at the factor plot of the molecules, these don't separate by their affinities towards the receptor. They separate by whether they're bivalent molecules or monovalent molecules. So what this seemed to indicate to us is that bivalency is a really important effect in making a specific cytokine for Tregs. Now, that was weird and surprising to us because we didn't really expect that valency would have such an outsized effect, that the affinity of the molecules really wouldn't actually matter all that much. So we've done a lot of work in trying to use a mechanistic model to look at multivalent interactions, and I'll show this for another application in a second as well. And this is an extremely simple model. So you have a multivalent complex. This could be a molecule that has 4 different IL-2s as these little circles. It interacts with its monovalent interaction kinetics, which we actually have experimentally measured. And then what we assume is that this kind of can become Velcroed onto the cell surface. So the subsequent interactions are adjusted according to the fact that this molecule is already on the cell surface, and so its local concentration is increased. And we found that this actually explains this data really well. So if you look at the correlation overall, it explains 80–90% of the variation in the data set. If you look at even subsets here, where we're looking at the correlation across all of the Treg responses or all of the T helpers, or we look at the correlation for a specific cytokine across all of the cell types, in every case, we're seeing that the multivalent binding model, even though it's essentially not fit at all, that it can explain the data overall quite well. So that indicated to us that it's really some sort of difference in interaction kinetics that can explain why bivalent molecules are more effective. What's nice about having a mechanistic model as opposed to dimensionality reduction is that we can actually simulate what should happen for new situations. So here's what we did is we optimized for regulatory T cell signaling, keeping the other cell types about constant. What you can see is that if we go from monovalent to bivalent, that bivalent is better than any monovalent molecule can do. And then if we were to make, say, a tetravalent molecule, we can have something that's more specific for Tregs than any of the bivalent molecules. And this just keeps going up, although making a molecule that has 100 IL-2s on it is not really experimentally feasible. So that's actually what we're doing now. So this shows 2 different forms of FC fusion that we've done previously, whether you do it at the C terminus or the N terminus. And then we've made a double fusion. So we have just 4 IL-2s on here. And this actually just requires doing both of the fusions that you otherwise would use. And then the multivalent binding model actually indicated to us that something that would be even better would be to use a dead IL-2. So something that only binds to the alpha receptor, but doesn't signal at all, and combine it with an active IL-2 on the other end. So these molecules basically get it to the regulatory T cells. These other molecules are the ones that have the signaling. They're less specific, but the dead version of IL-2 provides some of the additional specificity. So we're now exploring these, and we have some recent in vitro signaling, but it's a long process to actually checking that this specificity exists. Okay, great. So even in, in quotes, a simple system can, we can learn a lot about it by being able to explore what the patterns are in the data. Can we make this a little bit more complicated? So in our bodies, if you maybe got a vaccination, what you develop is a whole panel of different antibodies that are specific towards those antigen targets, the thing that you were vaccinated against. But these antibodies aren't uniform. Not all antibodies are made the same. And there's a real interest in being able to explore how antibodies are specific towards certain proteins or certain parts of, say, a virus, but also how they interact similarly or differently with the immune system. And that's because when we have, say, a mixture of antibodies that our B cells have created after something like vaccination or an infection, there's the upper part of these IgG molecules that binds to a certain target, be it a cancer cell or an infected cell. But then a lot of the protection that we derive from these antibodies arises from the Fc portion of the antibody, so the lower part here. And this interacts with essentially almost every immune cell type except for T cells. They interestingly don't express these Fc gamma receptors. And this has been an interesting system to us for a long time because, like we saw with IL-2, multivalency is very important here. So these immune complexes are recognized by immune cells through the fact that there's multiple antibodies bound to them. And it's a great system that's been really well characterized in these interaction kinetics. And so it's something where we've been able to build models of how there's responses to antibodies, either monoclonal or therapeutic antibodies, or the antibodies that we make ourselves. And we can explore that and then experimentally test how responses occur. And I don't think I need to motivate why understanding how antibodies work in the body is incredibly important, especially after the last couple of years. But we've mostly been thinking about this sort of from the bottom up. Like if we take a molecule where we know everything about it, maybe this is a therapeutic antibody against cancer, can we build models of how it interacts with the cancer cells themselves and then how it interacts with the immune system? But in a lot of cases, we have measurements, complicated measurements from people, and it'd be nice to have an approach where we can actually take these complex measurements and make them simple. So just to show you a little bit of how we've done some of the more mechanistic modeling, we've been working with some collaborators, immunology collaborators, that have this really nice synthetic system where this would be like a simulated cancer cell or a simulated virus. And they can take a BSA molecule, they conjugate TNP, which is just a tag, onto it. And then they can form these synthetic immune complexes. So this is essentially what the immune system recognizes as something that's foreign. And then we can use this to explore what's the effect of having few antibodies or lots of antibodies bound compared to the affinities of the antibodies, both towards the antigen and to the immune receptors. And again, we've used this multivalent binding model that has these two steps where there's interaction with the cell surface and then additional interactions that lead to these multivalent complexes. So this is showing some of the data that Anya and Falk have been able to collect, where this is looking at binding to one of these immune receptors in an in vitro synthetic system. And what you can see is that basically if you change anything, the data changes. So if you go from IgG1 to 2, you'll see a difference in the amount of binding. And they had really done a detailed study to collect all of this and sort of had to throw their hands up and say, well, okay, you know, multivalency helps for binding, but we don't really understand much beyond that. And so what we did is we showed that this really nicely matches this mechanistic model of cell surface interaction. But what can you do with an in vitro model of interaction when what you actually care about is in vivo responses? And to answer that, I'm going to show some of the data that's been collected to show that these interaction with immune receptors is incredibly important. So this is a TA99 antibody. It binds to melanoma cells. And this shows just how important interaction with the immune system is to immune protection. So this TA99 antibody, it's interacting with these immune cells, but these are lungs from mice showing the metastases with the melanoma cells shown in black. And you can see that there's really, there's no clearance of cancer cells when you treat with this antibody. Now, if you have another antibody that has the same amount of binding to the melanoma cells, but you just change its interaction with the immune receptors, you go from an antibody that has no effect to something that almost completely clears melanoma cells from the lung. And this has been modeled in a way. How people modeled this previously is that they took the affinity towards an activating receptor, divided it by the affinity to inhibitory receptor, and they called that this AI ratio. And the point of that was really that the AI ratio predicted how effective your antibody was. So we asked, well, okay, if we plug our binding model in between, can we do better in predicting metastasis? And it turns out, yes, you can. We can predict how effective your antibody is across this set of different antibodies, as well as knocking out different receptors or depleting different cell types. And then what was really nice about this, and I won't show all the details because this is now published, but that when we break open the regression part of the model, so the regression part really weighted how important each cell type was, that this depended on classical monocytes, which is a population known to be incredibly important for clearing melanoma cells from the lung. So the model was not only mechanistic, or it was not only predictive, but it also was based in the mechanism of how this works. So how can we think about this more complicated situation? We have some people, in this case, it's going to be HIV patients. They have B cells that make a whole panoply of antibodies of different forms. These are different in how they interact with the virus particles. They're also different in how they interact with the immune system. And we want to come up with some patterns here that might explain how effective, how people get different immune responses to HIV or how they progress in their disease. And the principle, so what's really nice is that there's a new field of system serology that's done a lot of characterization of these antibodies. And what they do is they isolate antibodies towards a particular antigen target and then measure the interactions with the Fc receptors. And it looks a little bit like this, where we have a bead that's identifiable. We conjugate it with an antigen. We use that to purify out antibodies of interest. And then we detect how these immune receptor interactions are different using this detection reagent, which is usually something like one of these Fc gamma receptors. And what we noticed with this is that there should be a tensor pattern in this data. So just for example, let's say you have no antibodies that interact with this antigen target. In that case, you're not going to have any signal across the different Fc gamma receptors because there are no antibodies to interact with in the first place. So we should expect that there's patterns across antigens for the different receptors, and that there's patterns across receptors for the different antigens. And that's exactly where these methods are going to be useful. So we organize this data in tensor form, where we have axis that indicates the different receptors that were quantified, antigens, and subjects. And there were also some separate measurements made of how the antibodies are different molecularly. And this wasn't antigen specific. So we're using a method called coupled matrix tensor factorization. And this just starts to show how we can integrate data across different data sets, even if they have different shapes. And this is, CMTF is again explaining the variance across the entirety of the two data sets. So we're not finding patterns that are specific to this data set or this data set. We're finding patterns that can explain all of the measurements that were made across both of these. And so we decompose this down, and then we can look at these factors and use them to explain what the patterns are. So just like PCA, we can look at the amount of variance explained with these different numbers of components. One of the nice things we saw is that if you compare this to PCA, we actually have the same reconstruction error for a decomposition that's about half the size. So we're explaining the same amount of the experimental data using just a smaller set of numbers. So that should make everything a little bit easier. One way that we checked that we could actually, that we were finding patterns that were meaningful in this data is we can leave out measurements and then see how well we can infer there's left out measurements. So this is doing that for a single measurement, and we do a little bit better than PCA in imputing these missing measurements. And then we got kind of ambitious and we asked, well, what if we remove a measurement across all of the subjects? This is something that PCA wouldn't be able to infer, because this would be like removing an entire column from your data. There's no way to figure out what that column represented. And in fact, because our method uses the antigen-antigen and receptor-receptor relationships, we can impute these missing values quite effectively, even when an entire column is missing. So now that we reduce down the data into its component patterns, we can see, well, do these predict whether someone has a viremia or they're progressing in their infection? How effective is their immune response to virus? And we do about the same as how people did before. And so that you might ask, well, why would you want to do this complicated reduction technique if you're predicting these things just as well as before? Well, the reason is, and this is a common issue that is important to keep in mind with building sparse models like what was used before. Sparse models like lasso or elastic net, they're very effective at prediction, but especially when you have strong correlations between measurements, you can end up with the model not being able to tell you what variables are important. So it's difficult to see each of the things that the lasso or elastic net model is predicting here to be important. So these are the component weights or the coefficient weights of those models. But what we did is we bootstrapped the model, basically asking, if you were to go collect a new data set, would this model be similar or different? And what we find is that the variables selected are completely different every time. So although elastic net models were really effective in predicting how people's infection was progressing, this just shows that we can't use those models to figure out which variables led to those predictions. And in contrast, when we do that with the CMTF reduced data, this is showing bootstrapping the component weights with the reduced factors. We see that we get very reliable relationships, like component 4 is very consistently associated with progression. So just to show some things that we learned from this. So again, like I mentioned, component 4 is associated with progression. And we see this really nice shift, not in the receptor affinities. So you can see that there's many components that have the same pattern of receptor interactions, but we see a shift between external antigens like GP120 towards internal antigens like P24. So this suggests that there's a antigen-antigen shift when you progress in your disease. And this is something that people had sort of piecewise evaluated before. Like there are previous papers that showed that P24 antigen predicts progression. But what we can see is that this is really a pattern that occurs across antigen responses, that it's not just P24 specific. And another really nice thing we were able to see is that components 1 and 5 predict complement response. And one of the parts of the complement pathway is lectin binding interactions. And actually this component that just helps in predicting complement response is really unique in having lectin binding interactions, even more so than its specificity towards different antigens, although it is quite specific towards the external antigens. So again, this is data that's much more complicated in having a mixture of molecules, and we're able to find the patterns in that data. So let's just make this as complicated as possible. Let's take a set of patients that have an infection. We'll try and measure as much as we can about them. We're going to take plasma and serum cytokines and gene expression, and then try and predict whether they resolve their infection in 5 days or they continue to persist in their infection. And this is a huge effort across a number of different clinical collaborators, including some collaborators at Duke that actually collected these samples, and then Elaine Reed and Michael Yeaman, and the rest of the investigators here have been really important at UCLA in being able to make these really widespread clinical measurements. And so again, we're going to use CMTF. We're going to try and find patterns across our 2 data sets, coupling them. because we want to find how people are different, both in their cytokine responses and in their gene expression. Just like before, we see how many components we need to explain the data. One of the unique things about when we're coupling a data set is that we can actually prioritize by scaling the data, whether we explain the cytokine data preferentially or we explain the RNA data preferentially. So this is kind of a choice in how we prioritize the two different data sets. And it's something that we have to make a decision about when we want to figure out which patterns are most meaningful in our data. So we did this and then asked, well, across different scalings or different numbers of components, what is best able to predict MRSA infection outcome? And it looks like it's about 8 components. And then what's interesting is that an intermediate scaling was best able to predict infection outcome. So this suggests that having patterns across both data sets is really important for predicting outcome. Just like before, we can then use these reduced factors to make a prediction about something. And here we looked at both whether someone persisted in their infection and whether there were patterns that were associated with different sexes. And you can see here that one of the things about dimensionality reduction is that it can end up with patterns that explain batch effects. So here we have 3 different patterns that mostly have to do with the fact that cohort 1 is a little bit different than cohort 3. Cohort 3 was collected about 2 years later. But because we can separate out those effects, we are able to see that these patterns aren't associated with persistence. And so we've essentially corrected for the fact that these patterns explain some sort of technical effect. What was really nice, though, is that CMTF, so these combined patterns, are better able to predict prediction or outcome. So they're more accurate in predicting whether you persist in your infection. And then we can break open the model that we have here and actually look at how these 2 patterns that are associated with persistence are related to the cytokine measurements and the gene expression measurements. So what was really surprising to us is that both of these components are strongly associated with IL-17 or a Th17 response. And that's been previously characterized as being protective in MRSA infection. But here we see a strong association with persistence. But one of the things that was enabled by analyzing the data this way is we could look then at the gene expression patterns. And while this represents a Th17 response, it seems to be associated with this real strong expansion of immature neutrophils, which is something that in some of the mouse models now, they've been able to characterize that when a tissue has a persistent MRSA infection, there is this Th17 response, and that is generally protective, but it's not protective in this case. So if your infection persists, you continue to get more and more neutrophils, but they're not matured, and then therefore they're not protective. So it's just an example of a pattern that we otherwise wouldn't be able to see, because it's very important that we were able to couple the 2 different data types. Okay, so what are some general lessons here? So methodologically, dimensionality reduction's super important. It's already used all over the place. I don't think I need to motivate that too much. But it really helps with visualization, reduction of the data. You can integrate different types of data, denoising, and you can infer what missing values would be. And it was really important here because sparse models, like we saw with the serology data, they are predictive and they can be extremely predictive, but they're not reliably associated with individual variables. They can be, but especially in the presence of correlation, these models can be difficult to interpret. And tensor methods aren't automatically better, but if you have data where there's this slice-specific variation, they can be extremely effective, even beyond something like PCA. And they're extremely effective in visualizing or separating these mode-specific effects. So we saw, for example, you can eliminate how is concentration different from time in my data set. Some of the things we learned using these is multivalency is really critical for IL-2 therapies. We saw this really simple then antigen shift that predicts HIV status in what otherwise seems like really overwhelmingly complicated data. And then there's this IL-17 transcriptional program that we're now looking at and experimentally validating that seems to predict MRSA persistence, so a lack of resolving your infection. And this is also associated, interestingly, with females, which is sort of backwards, or backwards from what our intuition was, because most persisters are males in our cohort and generally. So why aren't you using tensor decompositions? So just some examples of other places where these we know would be extremely effective is like in ECM microarrays, where you have ECM data by cell line, by drug treatment, by measured quantity. And we've actually started working with some people that are interested in finding patterns in this kind of data. You might also have measurements across cell types, subjects, and time. This, with the ability to make systematic measurements and more high throughput, this really comes up, this situation exists in a lot of different places. And I think these methods will be really important to help explore this data in the future. There's also reasons you might not be able to use these methods. So one of the things that we've struggled with is if you have cohort data over time, just a practical issue is that people come into the hospital at different times. So you're not able to align all of your measurements across the time dimension. And that means it's difficult to use existing tensor methods. So we're interested in making some methods that actually make it possible to have non-aligned dimensions in your data. We're also have been exploring how you can actually do this coupling in a larger scale. So this top row is kind of showing the analysis that we did previously. But now we're looking at things like how can we find patterns across different studies, as well as integrating different types of data. So one of the nice things about these tensor methods is that they're extremely flexible in how you design your data integration efforts. And tools to make this easier would make it possible to do it more regularly. And then finally, this is super busy, but really the point here is that we right now have these tensor methods that are purely data-driven. But I think an important part of this will be recognizing that we understand how some of these systems work. And so making predictions that use the benefits of dimensionality reduction at the same time as a mechanistic model will be really important to actually use the fact that we understand how some of these systems work. So with that, a lot of this work was in collaboration with a lot of really great folks, including Jamie Spangler out there at Hopkins, Elaine Reed and Michael Yeaman, who work in some of the clinical collections here at UCLA. This was funded through Vistera for the IL-2 work, and we're part of this NIAID FC killing consortium to make better antibodies. And I showed work from Cyrillis, who's worked on a lot of the serology. Jackson's been doing our MRSA infection work. Brian's been doing our IL-2 modeling. And Madeline has been working on this, this advancement to be able to deal with non-aligned measurements over time. So with that, yeah, thanks for your attention. I'm really happy to start taking some questions. And I'm really looking forward to the discussion throughout the rest of the day.